Today, we're diving into the absolutely revolutionary science of molecular diagnostics. This is a field that's not just about, understanding our own biological source code. No, it's now giving us the power to actually rewrite it. Ready to see how this all works? Okay, so picture this. Think of the entire human genome as a massive, massive book. We're talking 3 billion letters long. Now imagine you have to find one single tiny typo in that entire book, and that one typo could be the cause of a devastating disease. That's the incredible challenge that molecular diagnostics takes on every single day. And the tools it uses to do it? They are truly game-changing.
To really appreciate just how far we've come, we gotta go back to the beginning. Our whole quest to crack the code of life didn't start in some high-tech lab with supercomputers. Nope, it started with something a whole lot simpler: a garden.
You can really see the whole story unfold right here. Each discovery just perfectly sets the stage for the next one. It all kicks off way back in 1866 with Gregor Mendel and his famous pea plants, which first hinted that these little hereditary units even existed. Just a few years later, Friedrich Miescher actually isolates the stuff. He called it nuclein, which, of course, we now know as DNA. Then you fast forward 100 years to 1973, and bam, Cohen and Boyer create the first genetically modified organism. This was huge because it proved we could actually do stuff with this code. And this entire journey, all these breakthroughs, they all led up to 2003 and the completion of the Human Genome Project. The Human Genome Project? This wasn't just another scientific achievement in a long list. This was the starting gun for the entire modern era of medicine. For the very first time, we had the complete reference book, right? The full, unabridged blueprint of a human being. This incredible resource became the foundation for literally everything that came next. It let us compare, analyze, and diagnose diseases on a level nobody could have dreamed of before. So having the whole book is a massive step. But what if you just need to zero in on a single page to find that one typo? Well, that's where our first big tool comes in. It's called the polymerase chain reaction, or PCR for short. And you can basically think of it as a molecular photocopier. And that photocopier analogy? It is spot on. PCR can take a teeny, tiny, almost invisible speck of DNA, like from a single virus or a rare cancer cell, and just start making copies, billions of identical copies. This amazing process, it turns a biological whisper into a full-throated shout, which makes it super easy for our diagnostic tools to hear.
So how does it actually work? Well, it's a beautifully simple 3-step cycle. First, you crank up the heat to separate the 2 strands of DNA. That's denaturation. Then, you cool things down so these little guide molecules called primers can find and lock on to the exact sequence you want to copy. That's annealing. And finally, an enzyme called Taq polymerase gets to work, building a brand new DNA strand, making a perfect copy. That's extension. Now, you just repeat that cycle 30 or 40 times, and you get this incredible exponential explosion into billions and billions of copies.
So why does any of this matter to you? Well, the importance of PCR becomes crystal clear when you see it in action. It's the core tech behind the COVID-19 tests we all got so familiar with. It's used to find genetic risks for cancer, like the BRCA1 and BRCA2 genes. It's all over forensic science, analyzing tiny bits of DNA from crime scenes. It's even used for prenatal screening. It's, without a doubt, a true workhorse of modern science. Okay, so if PCR is like photocopying a single page, what if you could speed read the entire 3 billion letter book all at once? Well, that's where our next tool comes in, and it's called Next Generation Sequencing, or NGS. And this is where the scale just becomes mind-boggling. Think about it. The original Human Genome Project took over a decade and cost billions of dollars. Today, with NGS, we can sequence a person's entire genome in a matter of days for just a tiny fraction of that cost. This isn't just an improvement, it's a monumental leap forward in both speed and accessibility. And it's what's making routine genome sequencing a reality. And, it's not a one-size-fits-all kind of deal. There isn't just one way to speed read the genome. Different platforms are built for different jobs. You've got giants like Illumina, which are absolute powerhouses for sequencing whole genomes. And then you have this incredible new tech from companies like Oxford Nanopore, creating portable, real-time sequencers you can literally hold in your hand and take out into the field. It's all about having the right tool for the right job. The impact NGS is having on medicine is, well, it's profound. Doctors can now diagnose rare genetic disorders that used to be complete mysteries. In cancer treatment, they can sequence a tumor's DNA to find the exact mutations driving it, like in the EGFR gene for lung cancer, and then pick a targeted drug designed to shut that specific problem down. This, right here, this is personalized medicine. All right, so we've learned to read the genetic code with incredible speed, incredible precision. But what if we could do more than just read it? What if we could actually edit it? This brings us to what might be the most revolutionary tool of all, CRISPR. Honestly, the best way to wrap your head around CRISPR is to think of it like the find and replace function in a word processor, but for our DNA. It gives scientists the power to go into that 3 billion letter book, find one specific word, or even a single letter, that's causing a problem, and correct it with almost surgical precision. It's wild. And the way it works is just beautifully simple. A little molecule called a guide RNA acts like a biological GPS. It takes an enzyme called Cas9, which are basically molecular scissors, directly to the target sequence in the DNA. Once it's there, Cas9 makes a clean cut. After that, the cell's own repair machinery kicks in, and scientists can actually guide it to patch up that break with a new corrected piece of DNA. The potential here, it's almost hard to believe. We are talking about the very real possibility of correcting devastating genetic diseases like sickle cell anemia. It's also being used to create incredibly accurate diagnostics that can spot cancer mutations and to design totally new therapies that can precisely turn bad genes off or good genes on. So, where is all this amazing technology headed next? Well, the future isn't just about more power. It's about making diagnostics smaller, faster, and way more accessible. We're talking about a huge shift away from big, centralized labs to the doctor's office or maybe even your own home. And you can see the dramatic shift right here. On one side, you've got the old way, big facilities, waiting days for results, expensive tests. And on the other, you have this lab-on-a-chip idea. These tiny devices can run complex tests on a single chip, give you results in minutes from just a drop of blood, and they're designed to be portable and affordable. It's a total revolution. And there are 3 key technologies that are really making this happen. First is microfluidics, which lets us handle tiny amounts of liquid on a chip. Think of it like plumbing, but on a microscopic scale. Then there's nanotechnology, which gives us these ultra-sensitive materials for detecting what we're looking for. And finally, artificial intelligence, or AI, which is absolutely critical for making sense of the mountains of data these tools produce, finding patterns that no human ever could. And that, really, is the whole story of this incredible journey. From Mendel's humble pea plants to the unbelievable power of CRISPR, we have gone from just watching the rules of heredity play out to reading our entire instruction manual. And now we're actually starting to rewrite it. This all leaves us with a truly profound question. The science has given us a power that not long ago was just pure science fiction. So now, the biggest conversation is shifting from what can we do to what should we do? And that's a conversation that involves all of us.